Good afternoon, and today we are going to discuss about the fifth unit. So, transgenic animals. Okay, so fifth unit comprises of transgenic animals and what are the strategies involved in the production of transgenic animals, and mainly we were talking about, because in fourth unit we have discussed about the micro manipulation technology, in vitro fertilization, embryo transfer, and artificial inseminations and all. In fifth unit, mainly we are talking about the transgenic animals and what are the some examples for the transgenic animals, what are the methods involved in the production of transgenic animals, that we will discuss in the fifth unit. These are the topics to be covered in fifth unit, and this is the last unit in the fifth unit, and one more topic is left out, will be discussed in the next presentation. Okay, so we will go back to the slide. What is transgenic animals? Transgenic animals is an animal that has been genetically engineered to contain one or more extra genes in its cell. It now has a new inheritable property of some type. So that is a transgenic. It is genetically modified animal. Okay, that is, in simply we can say, genetically modified organisms or animals. Then what are genetically modified organisms? So genetically or modified organisms are altered artificially in DNA. So when you are altering the DNA, so then the genetically get changed. So this is the major important method for producing the transgenic animals. Like, it is created by adding a foreign gene or altering the base sequence within the existing gene, or deleting or turning off the some of the particular gene so that it can be genetically modified. Okay, next one, adding a foreign gene. So how can we add the gene into the animals so that it function properly, or if we are expecting particular functions from the particular organisms? So you have to insect a resistant plant, then bioremediation, then production of insulin, vaccine, herbicide resistant plant. So these are the applications when you are doing for when you are doing foreign gene addition. Okay, next one, altering an existing gene, that is altering the base pairs in the existing gene. So in existing genes in the organisms altered to make it produce or higher levels. For example, if you are taking a transgenic cows, so for getting a better or high amount of milk, that is it. Then, and also it influences the growth. So in case of deleting or turning off the existing gene, so it results in for example, that new gene can be inserted into the tomato DNA to give them a longer shelf life. So because now with that genetically modified tomato does not have suppose if a normal tomato, the lifespan is almost 2 to 3 days or more than 4 days, not more than 4 days. When you are talking about genetically modified or genetically deleted or turned gene into the tomato, that the life of the tomato will be extended. Okay, so next one, genetically modified animals. These are the plants. Now we are talking about the animals. It is micro injection, embryonic stem cells transfer, and nuclear transfer. These are the methods involved for the genetically modified animals. Okay, what is micro injection? So micro injections, first it is harvested, the eggs are harvested from super ovulated animals and fertilized in vitro. So in vitro, in the sense, we have to do in laboratory level. Okay, so first we have to harvest the eggs, then 200 to 300 copies of the foreign gene are injected into a recently fertilized egg. Suppose you are collected the egg and we have to inject the foreign gene into the egg and implant it into surrogate mothers. So you are taking a egg from the mother, okay, and injecting 200 to 300 copies of the any foreign gene, then put it into the surrogate mother. Okay, then a small proportion of animals born as, that is called as transgenic. Okay, see this is the micro injection process. When you are injecting in a microscope level, and you will get the transgenic plants or whatever it is. When you are talking about embryonic stem cell transfer, see, first we have to target the gene sequences to a specific site in the genome. Okay, alterations has to be made to the stem cells in the culture. Mutated stem cells injected into blastocysts, and blastocysts implanted into the foster mother. Okay, and transgenic animals are born. That is the embryonic stem cell transfer, and another, that is one. What is the method called? Micro injection. That is, okay, this is micro injection method, and this is embryonic stem cell transfer. Next one, see, this is the pictorial representation of embryonic stem cell transfer. First, that is, we have to collect the cells from the male animals, and we have to fertilize it. Then we have to inject the cells, copies of genes. Then it is 2 cell stage has to be taken, and it should be gone for 4 cell stage and 8 cell stage. This will go off, and 8 cell stage to blastocyst and fetus, and finally you will get the adult. So this is takes place in the human being. Okay. So next one is blastocyst contain pluripotential stem cells. Okay. So first you see that fertilized egg, it will go morula, and morula is changed into blastocyst. It almost approximately 120 cells are there, and about 4 days old culture we have to inoculate it in the culture, that embryonic stem cells. Okay, see, that is inner cell mass, and finally dissociate into the embryonic stem cells. Culture dish in incubator, and you will get that transgenic plants. Okay, so inner cell mass cells continue to proliferate indefinitely in culture. See, this is what. First we have to fertilized egg should be grown to morula, then blastocyst, then inner cell mass, then into the petri dish. Okay, we have to dissociate into subculture into pluripotent embryonic stem cells, and finally thaw, and you will get the embryonic stem cells. These are the stem cells which are used for the transgenic animals. Next one, nuclear transfer. There are 3 types of transgenic animals for the production. One is micro injection, one is embryonic stem cell transfer, and nuclear transfer. First one, remove the nucleus of the unfertilized egg and replace it with the nucleus of donor cell. That is the main here. Okay, when you are talking about nucleus transfer, so here everything taken care only in the nucleus. Okay, so remove the nucleus and replace the nucleus with the donor cell, and electric current is used to fuse the nucleus in the cell and trigger the egg to begin developing. Okay, so in the egg we have to remove the nucleus and replace the donor cells and give the electric shock, and the egg get started to fuse and to develop. Okay, the reconstructed eggs are then implanted into the surrogate mother. Then somatic cells can be cultured, subjected to the genetic manipulation in vitro, and then produce viable animals by means of nuclear transfer. So this is the method for nuclear transfer. See, genetically engineering animals, first we have to do, and udder cell was removed. This is the egg from that animal goat. Okay, then you have to add the nucleus removed from that old or parental goat. We have to add the donor cell, then it will change it into, that is the nucleus. Okay, nucleus from the other cells, it was new, and the udder cells was mixed, then developing the lamp embryo, and finally you will get the dolly, that is cloned sheep. Okay, so cloning procedures is, this is here. See that in a culture petri dish we have to add blastocyst, early embryo, fetus or adult. Okay, possible donors. These are the possible donors for the developing the cloned animals or transgenic animals. That one is, we can use blastocyst or early embryo or fetus or adult. Okay, then culture cells. So for example, blastocyst you can directly add to the cell or not going into the culture cells, we are directly into the cell, single cell, and it will be recipient egg, remove the nucleus and insert this egg and fusion, and you will get the implantation into a surrogate mother, and finally you will get the progeny. Okay, so Dolly and the transgenic clones. So Dolly is the first uh, produced transgenic animal. It was born in July 1996 and was introduced in the year same 1990, not in the same year. It is introduced in the year 1997 in the March month. Okay, so then later Wilmot and Campbell fused 277 adult mammary gland cells with 277 egg cells, but they achieved only 13 pregnancies and only one resulted in live birth, but that one offspring Dolly. Okay, so Wilmot and Campbell, they have tried in 277 eggs and they replaced the another with the 277. Out of 277, they succeeded only 13 pregnancies. Out of that, one is Dolly. Okay, next one, the scientist who is A is the Wilmot, Ian Wilmot, the Roslin Institute researcher, and B is the Dolly cloned sheep, which is a mother but no father. Okay. Next one, transgenic animal products. So what are the products from the transgenic animals that we have to take in care right now and that we have to discuss it. First one, human lactoferrin in cow's milk. That is lactoferrin, a protein found in animal milk that has ability to bind iron molecule. Okay, so that is in the initial presentation itself I have told you. So transgenic animals which give you the high much or very much yield or large amount of milk can produce. Okay, by altering the lactoferrin. Okay, lactoferrin is a protein which is found in the animal milk that has ability to bind the iron molecule. So the protein has strong antibacterial properties because it removes iron from the environment that bacteria need the growth. Okay, it relieves the iron deficiency anemia. Okay, transgenic code. Transgenic code, that milk contains protein normally found on the surface of the parasite that cause malaria. So because that which some of the milk which caused by the parasite and leads to malaria, the protein elicits an antibody response in test animals and could conceivably be used to immunize against malaria. That is the another important thing. Okay, next one, human hemoglobin from pig. So we can isolate the hemoglobin for the humans from the boar or pig. So transgenic pig that produces human hemoglobin, one day old, that is okay, 1 day old embryos received injection of 2 genes for hemoglobin protection and the embryos were then implanted to surrogate. Okay, see, these are the general procedure. First we have to do the we have to choose the types of method which needed for the transgenic animal protection. One is microinjection, embryo stem cells transfer or nucleus transfer. Okay, so for human hemoglobin, they have injected the 2 genes into the pig and the embryos were then implanted into the surrogate. Only about 5 in 1000 injections succeeded and only 3 pigs survived to the birth. That is the reason, because see, when you are talking about the Dolly, because it is the famous one and this is the first animal which has a transgenic and which was succeeded only that Wilmot tried 277 eggs with the 277 new nuclear eggs and replaced, and they have tried in the surrogate mother and they developed the Dolly. It only succeeded 13. Among the 13, the only survived is Dolly. Okay, so that is one negative side. The nuclear transfer technology results in high mortalities and low birth rates, because nuclear transfer technology which has negative feedback also, because which has high mortality and low birth rate when compared to embryo stem cell transfer and microinjection, and offspring are abnormally large size, undeveloped lungs and ill health, implantation problems are there. Okay, see, when you are talking about the nuclear transfer, we have seen so many animals which are infected, which have a disorder, genetic disorder, the genetic disorder because of this cloning or this artificial insemination or or in transgenic animals. It can be done by replacing one cell to another cell, one egg to another egg, or one gene to another gene. That is the reason. Okay, so and another one is the main drawback by use of nuclear transfer. Poor placental development are common because the placenta between the mother and the fetus, it should be very in strong. But if it is poor, then taking of food from the uh, offspring, from the parent, it's very difficult. Okay, then transgenic animals, some of the examples for transgenic animals, this is cloned kitten and this is cloned pigs. See, cloned pigs when compared to, see, nowadays so many things are happening. So I will tell you one best example. In India there are three varieties of sheep are there. In these three varieties of sheep, uh, they tried cloning or they tried the transgenic with South Africa sheep. They brought the cells from the South Africa goat or okay, and they implanted in our Indian sheep. And similarly for three varieties in India, there are three varieties are there. In these three varieties, they inoculated the nucleus, or they modified the genes, or they replaced the genes, whatever it is. The method is same, and obviously you will get the product as transgenic animals. So they tried in either one of them. I think most probably embryonic stem cell. So they tried and they succeeded, and the growth of the sheep is looks like horse, because it is larger in size, larger in height, and that the yield which is very high when compared to normal shapes. So these are some of the examples for the transgenic animals. Okay, and another thing, transgenic animals is this is a mute which is tried in horse, then transgenic endangered species. See, sheep, so that is what I told you. In sheep, they have tried so many things. In Kerala, they have one variety of sheep. In Tamil Nadu, they have another variety of sheep. And in, I think in Madhya Pradesh or Rajasthan, they have another variety of sheep. There are three different variety of sheep that are there, but all are looks like same in color, same height, and same resemblance. But the yield and the genes between these three is very varied, and they have tried in all these three ships with the foreign stem cells that is collected from South Africa. So they inoculated in these three ships and they get succeeded, but the yield is not that much in case of Tamil Nadu ships. That is a drawback. But they have succeeded in Rajasthan ships and another ships. Okay, and questionnaire why we are so that is about the transgenic animals. When in transgenic animals, we should know that what are the transgenic animals are there, what are the transgenic in what are the species they have tried. That has to be taken care of it. First one, some of the questions will arise into the in our everybody's mind. So do transgenic animals have direct benefit to humans? That is a question. The answer for that, DNA technology have been able to produce transgenic pig that produce human hemoglobin. Okay, so that purified hemoglobin from this animal can one day be used in place of human blood for transfusions during many surgical procedures. That is the reason. So in human globin, they have produced it from the pig. Okay, but till now they are not tried. Maybe they may purify the hemoglobin and they try to succeed in humans. That may happen in the future. Okay, next question, are there any other products possible from transgenic animals? Yes, because the research is currently working on enormous areas. Okay, the research work is currently progressing in methods to obtain compounds that may be used to treat emphysema, cystic fibrosis, and dissolve the blood clots. So these are the some of the question line, and which helps you that regarding transgenic animals. And next one, How about improving the quality of animals itself? That is a good question which comes in our mind. So that is, transgenic cows could one day produce higher quality of milk, one that is similar to human milk. Okay, so that cows produce the transgenic cows nowadays, that we know that transgenic cows are produce the enormous amount of milk, but one day it may happen or may not happen. So that is a research, right? So next one is chicken and cattle that synthesize the antiviral proteins which encourage the disease resistance in their bodies. Such animals would require less antibiotic therapy. Okay, so chicken and cattle these are synthesize the antiviral proteins which encourage the disease resistance in their bodies. Okay, next one, what are the risks of cloning? So that is very, because even when you are talking about monoclonal antibodies, we are uh, we are cloning the hybridoma cells for future use. So what are the risks involved in the cloning? That is, first one, reproductive cloning is expensive and highly inefficient. Then more than 90% of cloning attempts fail to produce viable offspring. So because the risks involved in cloning are, the reproductive cloning is very expensive, and more than 90% of cloning attempts to fail. Then more than 100 nuclear transfer procedures could be required to produce one viable clone. Okay, then higher rates of infection, tumor growth and other disorders. And last one, Australia's first cloned sheep appeared healthy and energetic on the day she died. That is the risks involved in the cloning. See, in Australia they have tried in the sheep for the transgenic, and they have succeeded. They succeeded in the transgenic animals, and it is the sheep is survived for some time. But on the day of she is going to die, she is very active when compared to the previous days. So that is the risks of cloning. Okay, and so from this presentation I have to summarize that transgenic animals, and what are the transgenic animals which are genetically modified, either nucleus or eggs or stems cells. Okay, so you can alter this cells from you can take the cells and alter in the sense, you can take cells from one species and replace the nucleus from the in their own organisms, and you can inoculate the new cells and check the transgenic animals. So there are three methods involved in the development of transgenic animals. One is microinjection, second one is embryonic stem cell, sorry for that disturbance. See, I will conclude that genetic cells which are produced, there are three methods. One is microinjection, second one is embryonic stem cell transfer, third one is nuclear transfer. A microinjection can be done in the microscope level, and embryonic stem cells which has a wide variety of applications, and when compared to nuclear cells, because nuclear cells have some negative feedback, and so embryonic stem cells will succeeded, and the embryonic stem cells which in there they replace the egg. See, we have get the egg from the one animal and inoculate it 200 to 300 copies of genes into the egg, and by applying the electric shock, then it will start to fuse and develop. Once it is developed to the particular stage, then you have to inoculate it into the inoculate it in the surrogate mother or surrogate animal. Then you will get the offspring or breeding. Okay, that is a newborn. That is a transgenic animals. Okay, so when compared to all these three methods, embryonic stem cell transfer method is more efficient, and we can easily achieve the transgenic animals. Thank you, and thank you for listening my presentation. Thank you.